It's 1 o'clock, so we'll go ahead and get started here. So thanks everyone for coming to my talk today on functional magnetic resonance imaging data. So I'm going to give a brief overview of what we'll talk about to start, beginning with an introduction to neuroimaging data very generally. Talking about the data cleaning process for fMRI data, which is called pre-processing. Then we'll talk about some common statistical methods of analysis for fMRI data, and conclude with a brief discussion. So this talk will be not too technical. We won't be getting too into the details of statistical modeling and such, but we'll go over a brief overview of fMRI very generally, and how statistics sort of fits into that. So why is fMRI and neuroimaging data very generally important? So one thing to look at, this trend in mental illnesses in the United States across time. So particularly among younger age groups, we have this increasing trend since 2008 through 2019 of an increase in the prevalence of mental illnesses. Okay. So you see, 18 plus, 18 to 25, all of these groups, an increasing trend. Neuroimaging, which broadly refers to the field of data collection and analysis methods related to brain measurements, can help us improve understanding of issues like mental illnesses and neurodegenerative diseases like Alzheimer's disease and Parkinson's disease. So neuroimaging as a tool is very useful for clinical practice and clinicians in potentially improving understanding of these issues and developing better treatments moving forward. Two of the main types of neuroimaging data are what we call structural and functional imaging data. So before we get into fMRI data, which is a type of functional imaging data, we'll talk about some of the common structural imaging modalities. So some of the most common ones, which you may have heard of before, are positron emission tomography images, or PET images. These use an injection of a radioactive tracer into the blood in order to produce a three-dimensional image of the body or brain. We also have CAT scans, computed tomography scans, which use X-rays to produce a three-dimensional image of the body or brain. And then we have magnetic resonance imaging scans, MRI scans, which use magnets and the varying magnetic properties of different tissues in the body to produce a three-dimensional image of the body or brain. So MRI in particular is useful in that it's a non-invasive medical imaging technique. It's not required that you inject any substance into the bloodstream, although that can be done to improve the quality of the image. It's not necessary, and it doesn't use X-rays, which can have harmful effects if repeatedly done across time. So there's no known long-term side effects of repeated MRIs. So this is a huge benefit in that it can still produce a very detailed image without harming the patient. And it's able to do this by generating a strong magnetic field and using radio waves to produce images of these internal organs and tissues in the body. So here's a depiction of an MRI scanner. So they're very large machines and very loud. That's not the best thing in the world, but again, nothing needs to be injected, and there's no long-term known effects from undergoing an MRI. So what does this MRI data look like when it's obtained from the scanner? So structural images that are obtained come in a three-dimensional array consisting of a large number of volumetric pixels, which in neuroimaging are referred to as voxels. Okay. So these voxels commonly for MRI data, and when we say MRI, that's often referring to structural MRI data, which is a single snapshot of the volume in time not repeated measurements across time, just a one-time image. For structural data, the voxels are typically 1 cubic millimeter in size. Okay. Although with increasing technology availability and improvement in scanners, they now can scan at the sub-millimeter level, they say as well, for a finer spatial resolution of the images. So this is an example of one slice of the array, how that looks with each square representing a voxel here. Talking about some common functional imaging modalities besides fMRI first, some of the most common ones are electroencephalography, EEG. EEG takes advantage of the electrical current that neurons produce when they fire, and it measures that by having sensors on the scalp placed like this. And so here's a depiction of EEG data. MEG data, magnetoencephalography, is very similar, but measures the magnetic field that results from the electrical field of neurons firing close to the cortical surface or the outer surface of the brain. So both of these are non-invasive techniques as well, although some limitations about them is that, especially EEG and MEG somewhat as well, they're mostly limited to the cortical surface of the brain, so very much the outer portion of the brain, but can't capture images in detail or as well for the inner part of the brain. Okay. Some benefits of them compared to fMRI though, they can be sampled with a very high temporal resolution, we say. So they can be sampled every millisecond, so you end up with 1,000 measurements per second, whereas fMRI, you can get one measurement about every 2 or 3 seconds typically. So a large difference there. But the benefit of fMRI is that you can obtain detailed pictures still in the very middle of the brain. So we're still interested in learning about other parts of the brain, not just the cortical surface, the outer surface of the brain. And fMRI allows us to do that. So here's a depiction of data from an fMRI study here. So we have the voxels, a single voxel here, for example, making up this entire image or volume. It's sometimes referred to as a neuroimaging. And we have repeated volumes obtained over time. So you can think of it as an extension of structural MRI, where we just keep repeatedly getting a picture of the brain across time. And this forms for each voxel in here a time series, a series of repeated measurements on the same thing across time. This additional data, this additional dimension to the data of time comes at a cost of lower spatial resolution. So the size of the voxels are larger than you would get for a structural MRI scan. So typically they're 27 cubic millimeters or 3 by 3 by 3. Although again, with improving scanners, the dimensionality of this is being improved as well, the spatial resolution of this.
Just a quick question.
Yeah.
What is it that's measured on a voxel?
Right, that's a good point, yeah. And yeah, right on the slide actually. So, so there's a few different options for what would you measure at each voxel. The most commonly used one is what's called the blood oxygen level dependent signal, the BOLD signal. So how this works is it measures the blood flow in the brain by taking advantage of the varying magnetic properties of oxygenated and deoxygenated blood. So by turning the magnet on and relaxing that, it can see the rate at which particles realign, and that gives a picture of oxygenated and deoxygenated blood patterns in the brain across time, right? And the idea being we're wanting to measure neurons firing or neuronal activity. The idea is that when neurons fire, they need oxygenated blood flowed to them. And then after they're done firing, the deoxygenated blood flows away. So this is a surrogate measure, we would say, for neurons firing, whereas EEG is measuring directly what we're actually interested in. So this is another difference between EEG and fMRI data. So talking about pre-processing of fMRI data, or data cleaning is one way to think of it, to get it ready for analysis and visualization. One of the first steps that we need to do is to convert the data from the frequency domain, which in neuroimaging is referred to as K-space, into the time domain, which is the more traditional realm, so to speak, in which we analyze data. So any time series data, repeated measurements across time, has a corresponding time domain representation and a frequency domain representation. The time domain representation, in this case, gives the strength of the signal as a function of time. It's more traditionally what statisticians would be comfortable analyzing for the most part, is data in the time domain. The frequency domain is an alternative representation of that data that encodes information about the frequency and phase information about the time series. So it's not that one point in K-space maps exactly to one pixel in the image. It's that one point describes many characteristics regarding frequency and phase information about the time series for the entire image. So some methods directly analyze data in the frequency domain for fMRI data, but most analyze it after it's been converted to the time domain. And this conversion is done using what's called an inverse Fourier transform. Some common issues that you encounter with collected fMRI data often need to be fixed or addressed before you can analyze it or create good visualizations. So one of the most common issues is motion correction or participant movement causing artifacts in the images. So here in this image is depicting on the left, a problematic image where there's some movement from the participant that caused some somewhat faint but white bands to be displayed across the image. This was caused by movement, which sometimes can be the participant moving, or it can be as small as just the participant breathing or swallowing. Those types of things can cause these artifacts, which would cascade issues into analyses if you fit this right into a model without accounting for it. So these can be removed using various statistical techniques like filters. And this is sort of like a interesting statistical problem in itself. How can we best represent the original image while removing these effects of movement prior to analysis? So this motion correction is commonly needed to be done. Another step that's necessary, especially if you have multi-subject data in which you want to conduct group-level analyses, is to standardize each participant's data to the same space. So if you want to compare across multiple people's MRIs or fMRIs, we need to warp or project their data onto a common space to facilitate comparisons. And the common space that's determined, there's a few very commonly used structural MRIs that research teams have developed in order for people to normalize or register to those images prior to analysis. One of the most common being the MNI space, they call it, which is a atlas. That's the name for this template, is also an atlas. It's an atlas developed by a research team at the Montreal Neurological Institute. So this allows us to compare across subjects, but also if you have repeated measurements on a single participant, say they might come in for a session this month and come in again next month, you want to be able to integrate their data together. They might not have been lying exactly the same in the scanner, and slight different orientations can cause the data to be oriented differently. So again, you need to align them to the same space in order to analyze it. Yeah.
So if I'm understanding correctly, are you talking about like one person's brain might be a different size, and that's the reason, or they might have swelling at one time and they don't have swelling at another time? Is that what you're looking at attempting to normalize?
Right. Yeah, that's exactly one issue that could be.
And because these boxes are an actual size, then if somebody's brain is twice as big, they just have many more boxes than the other do, as opposed to if you had measured based on percentage of distance from the center or something like that. So it's moving to something like that.
Right. Yes, exactly.
Okay.
And we might want to, for some studies, for example, we might want to explore activity levels in the parietal lobe. And so we want the parietal lobe to be in the same spot for everyone. We don't want it to be a different area that we're getting at when averaging across subjects. Another common issue, which I've seen myself after I got the data and was getting ready to analyze it and plotted it and noticed this trend, is that you can have what's called drift in the MRI signal. So here, the lower image, which is the problematic one, has drift, and that the signal or strength of the signal across time is increasing. And this can be such that even if you kept measuring for an extremely long time, the signal would just keep drifting on and up. But it's not that the activity level in the brain is continually increasing in that person. It's caused by imperfections in the MRI scanner itself and how it produces the magnetic field. So this is something that we want to remove. Often you can just regress this out as like a function of time to get a stationary time series here to get that ready for analysis.
How do you know it's drift versus that it really is?
Right. Yeah, that's a limitation for sure. And unfortunately, there's a lot of aspects with the pre-processing pipeline that are like that, similar to the artifact. How do you know you're removing artifact, or was it actually signal? Yeah, it's not for certain, for sure. So that was some of the main or most common steps of the pre-processing pipeline, but there's many others. Some being, for example, when you collect the data, you have all these measurements at each voxel in space. Sometimes the measurements can be quite erratic, so you might get quite extreme values. Like when I looked at, they called it DVARs in the pre-processing pipeline, but it's essentially z-scores for the values, standardized values of these voxel measurements across time. Some of them would be 8 or 9 standard deviations above the mean for just one time point, and then it dips right back down to normal values, which if you keep that in for traditional statistical models will cause a lot of problems. So you can do some spatial smoothing with various methods as well. That's commonly done. One way to think about that is you use a weighted average of spatially close voxels to smooth out the signal across the space domain there. So that's another example of a pre-processing step, is spatial smoothing. That also has, yeah.
Just wondering, is there any lack of what you might consider to be accuracy based on where it's located in the brain, like the more inner data? Is that harder to read or easier on the edges, or not? Is it all pretty uniformly
right?
accurate?
Yeah, so that sort of gets at actually another step in the pre-processing pipeline that I had to leave out for time here, is that when you acquire the scan, when the MRI scan does this, it gets one slice at a time, starting at the top of the head, gets a slice, and then moves down, gets a slice, and it goes all the way down to the bottom. So in that way, each slice has the same resolution, I guess, but there is like a effect of the difference in time in which those were recorded. So they have to be stitched back together to form the whole image. That's actually another step that needs to be accounted for in pre-processing as well. So in that way, I guess the resolution would be the same, but that is an issue with needing to stitch it all back together.
Thanks.
Yeah. Let's see. Okay, so yeah, so that was some common steps of pre-processing, which is not a simple endeavor in itself. But once you have pre-processing conducted for the fMRI data, you can then proceed to analyzing it or visualizing it. So one of the most common methods for analysis of fMRI data is to use the general linear model, so linear regression. In this case, and some of the research labs I was in, this was their default. They would always run this analysis and then see what else you could do after the fact. But this was the starting point. So for this type of analysis, if we had data on a single subject, Y is going to be the bold signal observed for a single voxel across time. So we're assuming we have capital N time points from this fMRI data, which typically ranges from 200 to roughly 1,000 time points for a single subject. So we have this voxel signal, and we're supposing it's a linear function in the way this is written of different demographic characteristics or treatment information, which can be encoded in this design matrix X here. So we're supposing we have P columns in this design matrix containing information that we're regressing out. And inference is conducted of beta here to see if demographic or group membership information is important in the voxel signal. Okay. And many methods fall under this general umbrella of the GLM. Some of the most common being ANOVA, multiple linear regression, and t-tests, with t-tests being sort of the favorite from my experience of what people like to use in this setting, where they simply look at a t-test to compare the average activation levels, say under two different treatments or between two groups. This is quite oversimplifying the data a bit. So probably some of the statisticians in the room are maybe cringing a little bit, worrying, aren't you glossing over a lot of details? And yes, that's the case. So there's issues of the data being temporally related across time. That should be accounted for in this modeling process. So you could do something like using generalized least squares, in which you account for the time series aspect of the data, although that has limitations, and that in order to use GLS, you need to correctly specify a structure on the error term, and that makes assumptions that might not necessarily be true as well. If you want to compare across multiple subjects using multiple subjects data, you ideally would need like a mixed effects model as well to account for subject or within group heterogeneity as well in the modeling process. So this is oversimplifying it a bit, but those would be the ideal routes that you would take in analyzing it with this approach.
And the predictors there, Andrew, are not time varying, right? They're just single.
Right, I think
single predictor value for an individual. They're not varying in time as well.
not typically, although you could have, some people do what's called task-based fMRI, where they have them do different tasks throughout the duration of the session, in which case that would change throughout time. Right, but yeah, most of the predictors would be like age, disease status, right? So that's one approach for analyzing fMRI data, which is the most common. Sort of flipping the role of X and Y, you can also have the fMRI data itself make up the design matrix or the predictor variable information and model, say, disease status or other outcomes of interest. This is also very common, increasingly common in literature, especially with machine learning being bigger these days. So some examples of this would be, like I've seen plenty of papers where people like to take fMRI data from individuals and use it to predict if they have Alzheimer's disease or not, based on their observed fMRI data. Many of these papers that I've read make you sad a little bit inside, because this is a very high dimensional problem, with the number of voxels typically being around 100,000. So for those with experience with machine learning, if you have 100,000 inputs you're taking into account in your design matrix, you can get very good at predicting things in your own data set, right? So I feel like roughly half the papers it feels like I've read about do some type of double dipping where they don't fully separate the training and test sets. So anytime you see a study that claims we can predict Alzheimer's disease status with 98 or 99% accuracy, I feel like it's nearly always misleading in their claim, and that they, like for example, one study I read, they first used the whole data set to decide which 100 voxels they would use, and then used those as predictors after separating training and test, and said, we got 98% accuracy. But it's like, well, we wouldn't be able to get that out of sample, right? So, but certainly an approach, if you properly separate your training and test sets, this could be a helpful approach to explore as well. Oh yeah, Jeff.
So,
yeah.
Andrew, are you familiar, since you mentioned Alzheimer's and that, are you familiar at all with the protein amyloid data that they think is a cause of Alzheimer's?
Right. Yeah, I've read into that.
Yeah, and this summer it came out. I think CNN had a story on it in Science News. That the original paper, I don't know if they used fMRI or what they used exactly, but they had kind of glued together multiple images, and now they've cast millions of dollars, right? And they cast doubt on any connection between that and it's interesting to me because we're always thinking of you talk about these ethical dilemmas and you think listening won't have it.
Oh, right.
But, they certainly do. And
yes.
Sort of using the training and test data set and making one and the same. Yeah, definitely feels that way also.
Right. Yeah, definitely. Yeah, I think I remember vaguely reading about that. And right, yeah, like you said, it's very costly. That it seems nice at the onset where, hey, look how well our drug does, but yeah, then you're costs you in the end. And of course, developing a drug that doesn't actually help people is not a great thing to do too, of course. So some analyses are done keeping data at the voxel level in this very high dimensional format. But what can be helpful is to reduce this dimensionality by aggregating or averaging voxel signals into what we call regions of interest, or ROIs, or what they refer to in fMRI analysis. So here's a depiction of this process where you have a whole volume that has, again, typically greater than 100,000 voxels, and then maybe you average it into these areas at each time point, which reduces the dimensionality of the problem, because now we can analyze data at the ROI level, but it also increases interpretability. Before, if you had a single voxel being important in predicting, say, Alzheimer's disease status, that's not as meaningful as if you had, say, the parietal lobe being implicated in Alzheimer's disease status. So by having it aggregated to this level can also facilitate interpretability and understanding the results for the analysis. So like my dissertation work always analyzed data at the ROI level rather than the voxel level. This also allows for, by reducing this dimension, many more statistical models are on the table and available for analysis. So that's a benefit as well. Yeah.
So is it like geographic, like nearest neighbor, like they're adjacent voxels or one voxel removed or something like that? Is that how?
Right. So these were much like the brain atlas that people register or project onto to get a common space. These were developed by research teams by using a large number of scans across many people to see, okay, here's where the parietal lobe tends to be by looking at structural MRIs. Here's where the like amygdala tends to be. And so they delineate the images by looking at a large number of participants' data that they've collected.
And then are things like kind of close to where it's supposed to be located or kind of far, or how do they measure like whether something is near a region, so to speak?
Right. So these are done, I guess this averaging is done after registering or projecting onto that same space to try and minimize how much it's off, but you're right in that it is still a problem that you might have some of the parietal lobe outside of the parietal lobe ROI, and that's a known problem or limitation.
Yeah,
definitely. Right. Yeah. Sango.
How that picture to your right look if you graph the CV, the coefficient of variation instead of the mean?
Interesting. Right, and CV is the ratio of the mean and the standard deviation. Yeah, I'm not sure. Well, this is this picture on the left or on the right here is using color just to delineate, I guess, the ROIs, but it's not showing like the signal strength.
But it's from averages, right?
It's from what?
It's based on averages.
Yes. So if you imagine this volume, we would get repeated measurements across time that are resulting from averaging the voxel value at each time point. Right. Yeah, so you could look at the coefficient of variation. That probably would be informative of things as well, rather than just looking at the average. Yeah, it's a good point. And to talk about the size of this problem, when I would analyze data, we would typically use anywhere between 10 and 50 ROIs, although some atlases, the definitions of creating these regions use as many as 200 or 300 ROIs still. So you still have a decently large problem compared to 100,000, it's much less, but even with 300 brain regions, that can still cause a lot of issues for some statistical models. So here's actual fMRI data from one of the data sets I analyzed, where I just picked out 4 brain regions. And here's the resulting time series across time for these. So you can see the green and red B and C brain regions at the top. You can see how they somewhat co-activate with each other. Their signals are related across time. As one tends to go up, the other one does as well. And even though the yellow region has a lower overall signal strength down here, it still seems to be co-activating similarly with the green and red region up here. But the blue region does not seem to be as related to the other ones here. So this idea of analyzing co-activation patterns in regions of interest or voxels across time is a whole large area of fMRI analysis that's referred to as functional connectivity. The idea being that potentially with some disease like schizophrenia, for example, maybe some regions don't co-activate like they normally do in healthy controls as well. Right.
When I see those two, it almost looks to me like an exponentially weighted moving average, where the time that gets factored into the average is bigger and less. I mean, it's almost like that blue one just doesn't move as much.
Right.
And when it does a big move, yeah, then it does a bubble, but not but not unless it's a big one. So it's like it's a bigger parameter incorporating more values into its average than the yellow, red, and the green. But that's just what I've seen.
Right.
Look at it.
Yeah, that makes sense. Yeah, sort of what I'm describing would be like the correlation coefficient between them. But yeah, there could be other measures of association to relate the regions as well. Right. So functional connectivity was being looked at as early as the 1990s for fMRI data. So formally, the temporal dependence of neurons firing, this activity of the neurons in the brain across time. And like I mentioned, alterations in functional connectivity have been implicated with various mental illnesses and neurodegenerative diseases. So like many of these studies I worked on were looking at the effect of, or the relationship between functional connectivity and schizophrenia. But others have looked at Parkinson's disease, Alzheimer's disease, depression as well. And like Paul was getting at, marginal correlation, looking at how the regions go up and down simply with each other, is one way to look at relatedness of the activation levels across time. But people do use other measures of association as well. The partial correlation coefficient is similar to the marginal correlation, but you can regress the effect of other regions out when looking at pairs of them. Coherence is a measure of association that's in the frequency domain. So you can think of it as analogous to the correlation coefficient, but calculating that in the frequency domain rather than the time domain. And mutual information, not used as much as the correlation coefficient is the default that most people use. But mutual information measures the amount of information that the two variables or two brain regions give about each other. That's based on mathematical statistics. So for the stat savvy in here, it looks at how much more information you get out of the joint distribution of two variables compared to just multiplying their marginal distributions together. The idea being if they're independent, those two things should be very similar. The more dependent there are, the bigger that difference will be. But marginal correlation and partial correlation are the most common. And these then are used in two sort of main areas within FC analysis. What are called seed-based methods, where you designate one brain region or voxel as a seed or reference region and look at the relationship. Oh, a chat question. Thanks. Yeah. Okay, thanks, Paul. Yes, yeah. Paul's talk is on EEG, I think, right? Yeah. So yeah, it would be very interesting as well. So for seed-based methods, we designate a reference region or voxel and look at the relationship of all other regions of interest or voxels with that seed region. So limitations of this approach are that there's many ROIs to choose or designate as your seed region. You might not have selected the right one to find the results you were looking for. And you could look at setting every single region as the seed region, but then you end up with many, many statistics you're looking at, a multiple testing problem if you're getting into hypothesis testing. So that's an issue as well. Graphical modeling, which is the methods I developed in my dissertation were all related to graphical modeling, works by commonly constructing what we call an undirected graph consisting of nodes or vertices, which represent the brain regions, and edges connect the vertices or brain regions in the graph if they're related to each other, if they're related enough. And that's where the different measures of association can be used to construct a graphical model. Oops. There we go. And then within graphical modeling, so we've gone into functional connectivity analysis, and then within graphical modeling, the most common approach is to use what's called a Gaussian graphical model. So for a Gaussian graphical model, it proceeds under an assumption of normality for the signals observed across time, which may or may not be reasonable. And under this assumption of normality, or a Gaussian assumption, you can use the inverse covariance matrix called the precision matrix to determine if nodes or brain regions are connected or not. So zeros in the inverse covariance matrix, called the precision matrix, correspond to brain regions being conditionally independent. So this would tell us that between brain regions A and D, there's a 0. So notice there's no connection in the corresponding graph here. And this relates or corresponds to the partial correlation between A and D being 0 as well. That's what the inverse covariance matrix tells us. So this would be a way to use essentially the partial correlation coefficient to determine your graph. You could use the regular covariance matrix, and zeros would tell us if marginal correlations between the variables are 0. That's commonly done as well. So wrapping up with some discussion here, there's a lot of ongoing challenges with fMRI analysis and pre-processing. So one of the biggest being, which was concerning to me as I learned more about this in analyzing this data, there's not a consensus in the steps of the pre-processing pipeline. So even within, say, one university, you might have multiple research labs or teams working on fMRI data. They don't necessarily use the same pre-processing steps. Depends on the principal investigator. They each have their own preferences, and they even, in my experience, switch it up study to study. As you learn more, sometimes you see, which is concerning, consideration of, say, 15 different combinations of different pre-processing pipelines, and then look at them all to see which one had the most interesting result. Yeah. So again, getting at John's issue with, is that good statistics, right? Probably not. You need to specify that ahead of time, right? So it's not supposed to be done, but I feel that it is, that idea of trying different pre-processing pipelines. So this has caused somewhat of an issue of reproducibility with fMRI studies or integrity of the results. This lack of a consensus on pre-processing pipeline also has caused issues with integrating data across multiple studies. So maybe you have 3 different research teams explored fMRI data and its effect or relationship with Alzheimer's status. You want to combine that information to get a larger signal, a larger amount of strength. But if they use different pre-processing pipelines, that process needs to be done carefully and causes complications. And I don't mean to be doom and gloom about this, because there's a lot of people who recognize this as being an issue as well and have done, entire research studies on what are good pre-processing steps to do, and here's how you should do it. Like Johns Hopkins University has some professors, Dr. Martin Lindquist, who's done some great work on calling out this issue and proposing solutions to selecting a pre-processing pipeline. Okay. Other issues, which we've already mentioned as well, the high dimensional aspect of the data. So for example, when I analyzed this sometimes with 16 gig of RAM on my laptop, you couldn't fit the data. You couldn't fit fMRI data on it because it's so large. So I had to piece it up, analyze data, or pre-process it for one subject at a time, use creative challenges like that. Or fortunately, Minnesota, where I went to grad school, had a supercomputer that you would use to analyze data as well. So these limit how you can analyze this type of data. The temporal and spatial dependencies, again, that necessitates more complicated statistical models to properly analyze this data. And also, a known problem in fMRI data is that it's very noisy. When you do have a signal, it's small relative to the size of the noise in the data. And so this can be addressed with having multiple participants, a larger number of participants, but again, that gets more costly as MRI data is expensive to obtain. And so wrapping it up, main takeaway, even though it has limitations and there's challenges, fMRI is a powerful tool that we can use to improve our general understanding of the brain and specifically mental illnesses and neurodegenerative diseases that may be related to this neuronal activity. And so applications of it are continuing to grow. People are improving methods and pre-processing steps, and it's also getting less costly, making it more widely available. So thanks. And we have some time for questions. That's great. Hey, John.
So you talked a lot about sort of that decision making on the pre-processing. Has maybe John Hopkins has done this, but has anybody actually done like a sensitivity analysis where they've taken the same data set and they've done maybe 10 or 12 commonly used approaches to pre-processing to see how much the results vary based on that?
Yes, exactly. Yes, that's been done right. I would say a lot. I read some papers when getting this together where they showed, for example, if you did too much spatial smoothing, if you over smoothed and you did a functional connectivity analysis, you could find more associations. And right, similar things with the other steps as well, like motion correction, how overcorrecting can affect the analyses as well, right? Yeah.
It almost feels a little bit like ecological fallacy, right? Where the unit of measurement at which you do the analysis, if you overly smooth, like if you look at ecological things on a watershed basis versus looking at it at a location basis, you get completely different correlations and associations.
Yeah. Which is concerning because right. So yeah, there's challenges definitely with the pre-processing. Mm-hmm. Yeah, something.
You know, if people have moved from parametric to semi-parametric model.
Right. So reading into this as well, right, parametric modeling makes a lot of assumptions about the data. So some feel very strongly that you should only use non-parametric methods in modeling fMRI data because of making less assumptions, right? So yeah, that's definitely done as well. Mm-hmm. Right, which I'd be an advocate for because it's always concerning when you see that they assumed it followed, say, an AR model, autoregression model, and say, we took care of the temporal dependencies, but it's a very strong assumption to make about the data, right? Any other questions? Thanks. Thanks everyone for coming.